begin our study with a little background on what it is we hope to achieve. The contents within our picture are the main focus, and I think that's important to remember. We hope to assess various microbial communities within the picture, as well as any effects they may have on the functions of the picture. So I think it's important to ask questions like, is there a relationship, in fact, between species diversity and function? And if there is, what effects and roles do these microbes play within our ecosystem?
Our study is significant because microbial communities are vastly understudied, especially in aquatic environments. And by understanding microbes in water systems, we can predict how various dilemmas may affect their communities, such as like global warming. Okay. We're using the northern pitcher plant for our study. This is a plant that has leaves that come out of the ground and open up to form a pitcher. So when rain falls in, plants go in, insects go in and drown. So they're carnivorous, and they have these tiny hairs in a downward-facing array, I guess, that when bugs try to crawl out, they get stuck and they drown. So they lure prey in with their nectar, sweet scent, and the unique coloration. The vertical shape of the plant is the key part to it. They have these transparent windows that fool flying insects into where the true egg really is. The microbes inside the pitcher secrete digestive enzymes, and then there are glands around the bottom that absorb the nutrients from the liquefied prey. These enzymes are our main focus because the rate can be used to indicate the change in microbial community. Nitrogen and phosphorus are provided from the decomposition of the liquefied prey, which is usually like ants, beetles, flies, wasps, slugs, and any organic material as well.
So when we talk about the habitat of the pitcher plant, I think it's important to first understand where it thrives, and it's unique in that it thrives in these nutrient-deficient environments, such as a swamp or a bog. These areas are usually very acidic in their soil, and they also lack nitrogen and phosphorus, which are essential for most growth as far as plants go. So, as Katy said, they rely on other organisms to provide them with nitrogen and phosphorus. These areas are also susceptible to submersion, where they're rising and lowering tides. And luckily, because these areas are usually hard to come by, our research led us to the Schultz farm and this bog where we did most of our research. Very serene, very quiet, very nice for a paddle in the morning. So to begin with our study, we went out into this bog and we selected 16 unopened pitcher plants. Unopened is important because this is a sterile environment for microbial communities to develop in. And we could start basically from zero and watch all of the activity as time progressed. Once the plants opened, we added our own version of rainwater, which is basically autoclaved mineral water that we brought from our lab to emulate rainwater to act as a growth factor for these microbial communities. The sampling took place over a course of 21 days. In the beginning, we would paddle out very nice in the morning. Can't believe we got paid to do this, but it's awesome. So we came out there and we collected liquid samples from our pitchers, as well as temperature recordings and pH levels. Once we collected these liquid samples, we put them on ice and brought them back to the lab for further processing. Pictures of myself on the field and some necessary work.
Once we made it back into the wet lab, each sample is separated for further processing. So one we put in the freezer to head back for DNA analysis, and one we used in the lab as we are. 3 enzymes were measured, so each day we had 64 samples total. And these enzymes cleaved and added substrate into 2 pieces. And one of the pieces has a yellow coloring to it, and so we can use a spectrophotometer to observe and record the wavelengths of these to see how they've changed over time. There's just a picture of a little lab work. So we decided to talk to you guys about AJ's favorite plant, the golden child. Here's a nice little action shot of a bug inside one of the plants. And this pipette is what we take out of each pitcher. And you can kind of see like little things in there that we pull out each day. Here's a graph of the golden child's enzyme activity. And I don't know if this pointer was going to work yet, but you can see that ACP is definitely the ringer of leading us off in how much enzyme activity we have. That doesn't mean that VG and NAG are inactive. They're just moving at a different rate.
So as far as early concerns go, I'd say we haven't had too many. Weather has obviously been a problem some days. One day it prevented us from going out in the field because thunder and lightning are not great when you're paddling in an aluminum canoe. I think you can imagine why. A couple other problems we ran into were pitcher rot and predatory insects. Pitcher rot occurs, as you can see down here at the bottom, pitcher rot occurs when these plants take on too many organisms at once and can't digest them properly. So as a result, they begin to degrade and eventually die, which turns into this brown mush you see at the bottom. At the top you can see some first and second year plants that are nice and healthy. Unfortunately, in the beginning of our study, we lost 2 of our plants, rest in peace, plants 2 and 9, to a predatory insect, most likely a wasp or a moth because they have very strong jaws. They bore a hole into the bottom of the plant and drain all the nutritional content out of it, eventually killing the plant. But besides this, I would say our research has been picture perfect so far.
And while we are sad to leave next week, the remainder of our labor is in the lab back at CMU, where we'll do our DNA analysis for the pitcher plants using PCR, D2GD, and sequence-based identification.
That is that. This is what we have done so far, and this is what we still have to do. So we have been very blessed to have the opportunity to be here, and we want to thank PCCI so far. And we look forward to showing you what we present in September. All right, thank you very much.
I guess, does anybody have any questions before?
Questions? Yes.
So that the enzymes are being produced by the plant or by microbes within the plant?
The bacteria within the plant. That was something that we were confused with when we first began, but yes, they depend totally on the microorganisms within.
And you're using autoclaved mineral water, you said, but rainwater would have probably microbes. It wouldn't have stuff in it, wouldn't it?
We just use that to start them off.
We used it to start, to emulate, and then once they were open, we let the rainwater and the natural processes take care of it from there. So it was just an initial startup. So we could start early, we could start from day 0, because once you open the plants, there's nothing in them, totally